So we're now going to move on to the last part of today, which is a talk from Thomas Keane. So this is the final of the webinars. Just share my screen. Okay, so the webinar today is on the application of FAIR principles in the CINECA project. So like with the other sessions, the webinar is being recorded. So we hope that is acceptable to everybody and will be made available. Just to put it into context for people, a couple of extra people joined just for the webinar. This is part of a series, a CINECA How FAIR Use webinar series and hackathon. So the webinar series ran from January to now, covering many different aspects of FAIR, general principles, as well as applying FAIR to cohort data, software tools, and training materials, and then the ethics and ELSI considerations about fairness. And then obviously it's the ending with this hackathon that's happening today and tomorrow. So today's presenter, I'm very pleased to welcome Thomas Keane. So he is an honorary professor at the University of Nottingham. His current position, his main position, is team leader of the European Genome Phenome Archive and European Variation Archive at EMBL-EBI. And so he does all the strategic planning for that. He's also very involved in the Global Alliance for Genomics and Health. He's involved in the steering committee and co-leads the large-scale genomics work stream. And that work obviously aligns very closely with the CINECA goals. So before joining EBI as a team leader, he led the Sequence Variation Infrastructure Group in the Computational Genomics program at the Sanger Institute, and his research interests are in using genomics technologies to understand biological processes. So he's also a scientific lead for the Mouse Genomes Project. So with that, I would like to hand over to Thomas, and we look forward to your presentation.
Great, thanks, Nikki. Thanks for a really nice introduction. Let me just get my screen set up. Let's see. There you go. You should be seeing my slides.
We do, yes.
Great. And yeah, okay, great. So I'm going to try and I guess talk about the CINECA project, in particular how the CINECA project is trying to address the FAIR principles and implement the FAIR principles. And I'll give some brief-ish kind of background about the CINECA project, what it's about, what we're trying to do, and then try to kind of step through, you know, the various ways in which we are trying to sort of further the fairness, I guess, of the human cohorts that are participating in the project. And I'll try to give some examples of the various initiatives that are happening across the various work packages, to give you a sense of, how we're trying to really move this forward and really get, move the kind of the whole standards environment, but then also use CINECA as a driver for the standards that are international standards that are emerging, and then fundamentally how we're trying to make the participating cohorts more fair, essentially, and use them as exemplars then for other sort of international kind of initiatives that are happening around cohort interoperability. All right, okay, so I'll just give some initial context. I mean, the kind of global sort of context, how we started with the CINECA project. I mean, where the sort of ideas were sort of coming from when we were sort of initially planning the project. I mean, we've seen the emergence of, over the last 20, 30, 40 years even, of many sort of really longitudinal research cohorts where, human populations have been studied for many years, and cataloging different traits and the occurrence of diseases, and then ultimately trying to figure out what are the associations, what are the environmental factors or genetic factors that are influencing those different outcomes and phenotypes. I mean, the sources, I mean, there are many different types of human cohorts out there. I mean, we've got many of the large-scale biobanks. In Europe, we've got the BBMRI-ERIC whole sort of infrastructure, which is tries to unify all the different biobanks across Europe. I think we're increasingly seeing, from national healthcare, starting to see clinical cohorts that are really being, made available and being studied for research through research studies. And I think it looks like, we're going to be getting the vast majority of the genetic data that we get, the human genomes fundamentally, the human genomes that will be sequenced are going to be coming from healthcare initiatives, and the hope is that a good proportion of those will be made available for research, to really get value from those cohorts, as well as delivering obviously sort of more immediate clinical outcomes to the healthcare. So one of the initiatives that's emerged in the last sort of 3 or 4 years is called the International 100K Cohorts Consortium. This was initially started up by NIH and Wellcome, and has gained more momentum since then. And that what they've been really trying to do is sort of map out, where are the very largest human cohorts. So they normally aim for sort of cohorts over 100,000 participants, but there are cohorts that are more specialized into specific, underrepresented populations that I think they have been included in this map. So they've made probably one of the first sort of global sort of pictures of large-scale human cohorts around the world, and that's the kind of initial setup phase for the IHCC. And really where they want to get to, I think, into the future is really what CINECA is trying to do for our much smaller number of cohorts, really trying to get the sort of interoperability with those cohorts in terms of accessing the data. How do you be able to use it, cross-compare, cross-analyze the data, to be able to answer questions that, by combining the cohort data that you couldn't answer by just, individually carrying out that corresponding analysis on individual cohorts. And this plot here, I mean, really gives a sense of why you want to be able to study ever larger cohorts, human populations and human cohorts. I mean, this is a slide I slightly borrowed from Rory Collins from UK Biobank, and it's really just showing that the resolution that you start to see, that the patterns that you start to emerge. This is looking at the correlation between age and blood pressure, and just as you go to much larger you collect data and phenotype data from larger populations, you just start to get these much clearer and much more higher resolution sort of relationships that emerge, as you're studying and looking at cohorts of larger numbers of individuals. And this is really essentially the potential essentially that that there are associations, genetic associations that we could potentially be able to discover if we were able to do study cohorts on a larger scale by combining the data and be able to cross map between them and be able to cross analyze. And fundamentally how you achieve that is to make the cohorts more fair. If you applying the FAIR principles, that is one way of enabling this to happen. So this plot here is just looking at genome-wide associations, the number of genome-wide significant SNPs that are discovered. As you study cohorts of larger numbers, and in particular, this is for common disease, where you really need to have the sort of cohorts in the tens of thousands to be able to see many of the associations. And then finally, just thinking about rare disease. I mean, the only way that what is a rare genotype, what is potentially a mutation or variant that could potentially be the source of a particular rare disease, is if you have a really comprehensive catalog. I guess this is sort of on that kind of genetic data theme again, is if you have comprehensive catalogs of common variation in the population. And one of the things the interesting points that having attended some of the initial IHCC meetings is that when I talk about cohorts, I mean they come in all shapes and sizes. There is just this huge heterogeneity, and that there's a really famous and big cohorts like UK Biobank and others that are really going into the kind of generating large volumes of genetic data and being able to do all these really interesting associations with that data. But there are many other cohorts out there that have been operating for 20, 30, 40 years, where genetic data is just not something that they could actually, they have the resources to generate at this point, but with the reductions in cost in sequencing and other associated technologies, proteomics technologies, you can see that it will be possible in the future, hopefully in the not too distant future, to really be able to study these cohorts and generate multi-omics data across many of these cohorts. Okay, so the CINECA project. So what are we trying to do essentially? I mean, ultimately, the mission statement is we want to sort of enable population scale genomic and biomolecular data to be accessible across international borders, and really to accelerate research and ultimately try to improve the health for the individuals resident in the across the continents and the countries that are participating. I mean, we really want to enable sort of the, really get to the point where we can enable sort of personalized medicine, enable novel discoveries by being able to combine across these participating cohorts. Some of the key features of the CINECA project. So we have 10 participating cohorts. As you look at that IHCC map, there are many more cohorts out there, and even cohorts in the world, but we have to start obviously with a sort of fixed number that we can actually tangibly work with, and obviously in the boundaries of the funding that we have. We've got about 10 cohorts. It's about 1.4 million participants across all of those cohorts. What's interesting about the project is that the cohorts span 3 different continents. So we've got the Canadian cohorts, the European cohorts, and the H3 Africa BioNet participation. So this makes it a somewhat unique project. I don't know, there's not that many other projects that are trying to do this cohort interoperability across multiple continents. I think we want to have, ultimately we want the CINECA project to be a driver for the federated analysis paradigm. What we're seeing, especially when you talk about, say, genetic data or multi-omic data coming from healthcare, is that data is probably not likely to leave those particular jurisdictions or those countries where that data is generated from. So we really see that the federated analysis paradigm is going to be, a sort of key enabler or key mechanism through which cross-cohort analysis will be done. So we're really trying to deploy these emerging standards, in particular some of the emerging standards from the Global Alliance for Genomics and Health, and trying to implement and deploy these with our participating cohorts to be able to demonstrate through our research and clinical applications that the federated analysis paradigm is the right. Well, we think it will be the right way to do this cross-cohort analysis, and will comply with the ELSI requirements. So we do want CINECA to serve as a driver for some of the international standards and the various participating cohort infrastructures and cohorts that are participating in the project. In particular, we've got a really close link with the Elixir infrastructure, and I'll come back to that in a few minutes and when I talk about more details about the FAIR implementation. We are deploying and implementing many of the Global Alliance for Genomics and Health standards, and really providing some of the very first implementations for some of those standards. One nice example is the researcher passport standard, which was only approved sort of late, I think it was late 2019 in GA40H, and I think it was in the first half of 2020. We, through the CINECA project, we had the very first sort of implementation of exchange of that passport standard, that researcher passport standard between Elixir and the EGA and the CanDig infrastructure in Canada. All right. Yeah. So I guess what's the starting point for CINECA? And we are, realistically, the whole cohort environment is completely heterogeneous. You've got cohorts that are funded in different ways. They're based at different institutes. We're collected through different consortia. Some of them are like highly active. Maybe some of the cohorts now maybe have run out of funding and become more static. There's custom formats, there's custom databases for representing and storing the data, but also the metadata describing the cohorts. There's different rules, different criteria, different systems, forms for getting access to the cohort data. This is, essentially every possible level of heterogeneity you could possibly think of, you will see across when you try to interact or get access to the human cohort data. There is hope here. I've talked about there are international standards emerging, and this is probably only in the last sort of 5 to 8 years, perhaps. These are really there's a real focus on developing these international standards. And as I said, the core goal for CINECA is to try and enhance the fairness of our participating cohorts by through supporting deployment and implementation of many of, well, as many as possible of the GA40H standards and some and other non-GA40H standards, and really show that those standards, if we can implement them at scale and then get some really good convincing research and clinical applications in the project, and use cases that really show these standards working at scale and delivering sort of practical, well, answering relevant research questions as sort of exemplars. So what are the challenges that we are trying to address through the CINECA project? So first one is, you can link these directly to the sort of the FAIR acronym here. The first challenge is the federated data discovery. So just finding, how do you find cohorts of interest that have collected metadata of that you're interested in, maybe that that the data use criteria are consistent with the type of research you want to carry out. Maybe you want sort of healthy controls, or you want cohort participants with particular disease types that have collected certain types of assays or measures, and that is probably the sort of first sort of fundamental challenge around cohort fairness. The second challenge is all around sort of data access and accessibility. So this is how this whole area of how do you actually request access to that cohort? How do you authenticate yourself? How do you get authorization to access that cohort data? A lot of this is quite manual. Well, it has been quite manual until quite recently, and probably still is, involving sort of paper forms and email chains, and really trying to see if we can use technologies and standards to really kind of accelerate that process. So, to really kind of make it possible to get access to the cohort data faster, with all the suitable controls as well. The next challenge was really around the cohort level metadata. As I said, these cohorts, many of the cohorts are collected at different times. They're using different coding schemes. They're using different ontologies. They're using different ways of storing the metadata, different measures that were collected, using different vocabularies. So, this whole harmonization challenge, is really fundamental to sort of enabling federated to enable the cohort discovery. And then challenge four. This is around the sort of really showing the kind of federated analysis. We have a set of use cases that we want to work with, and are really showing that this federated analysis paradigm, you can actually do research and healthcare/clinical applications and answer really relevant questions through this federated analysis paradigm. And then I guess, behind all of this, because as I said, we're working with cohorts across 3 different continents. We really have to look at the sort of ELSI framework and the ELSI regulations that are operating across these different jurisdictions, and trying to sort of look for the gaps and try to map where there is inconsistencies. And, I think that will be really understanding that landscape, and studying the sort of the recent regulations. GDPR is obviously the one example that's very recent, and that's the interpretation of GDPR is still being understood and tested, and what that means for research, human cohort research. So the structure of CINECA, we've got a set of data sources / cohorts. There's a listing here of the participating cohorts. We've got healthy longitudinal cohorts. We've got disease-specific cohorts, and we've got pediatric cohorts. It's a whole mixture. We didn't specifically target one specific disease type or one type of cohort. It was really we really got a sort of a bit of everything here in these different cohorts. And yeah, I think you've got the let's see, I think you've got the Canadian, no, there's no ordering here, but we've got, there's multiple cohorts in Canada, quite a few in Europe, and then obviously H3 Africa is the African cohort. And there's a mixture of different data types. Many of them got genetic data. I think most of them have got some form of genetic data of various types, and it's really the job of our research and clinical applications are really trying to look at which of the cohorts that would be appropriate for their type of federated application. And there's some quite interesting applications that are being developed through the project. Our work package 2 is all about the accessibility. So this is all about the the authentication, this whole authentication authorization business, and trying to get request access to the cohorts to be able to do the particular research you're interested in. And I guess the three sort of key infrastructures here is the Elixir AI, the CANDIG infrastructure. CANDIG is a cross, it's a federated infrastructure in itself, cohort infrastructure in itself in Canada, essentially. So CANDIG in itself has got participating institutes and cohorts, and it's trying to do that linkage across them. So it's, in some ways, it's the equivalent of a similar sort of role in terms of what the Elixir AI is playing. So we're trying to get linkage between the Elixir AI and the CANDIG AI, and then trying to get A3 Africa Bionet then connected into this kind of sort of network of authentication authorization infrastructures. Work package 3 is all about the cohort level metadata representation. I said, as I said earlier on, these cohorts are all collected at different times, different coding schemes, and the only possible way that you'll be able to actually ask sensible, query across these cohorts and actually get sensible answers back is that if you've got some sort of mapping across the metadata between the cohorts. And this is really it's a really key sort of part of the project that I don't think probably many of the other parts of the project couldn't succeed, I think, if we don't have this this metadata mapping model between our participating cohorts. Work package 1 is all around the federated data discovery. I've talked about this. I guess it's really kind of looking at sort of discovery by phenotype, genotype, and data use, and there's different levels of complexity of different cohorts or different queries. And we're really trying to align here with things like the Beacon network, the Elixir Beacon network. I think using things like the data use ontology to be able to codify, how you can actually, what's the appropriate types of research for particular cohorts in a way that doesn't involve, I guess, sending emails and or going through a long application process. You want to be able to see straight off, what is the type of research you can carry out on this particular cohort. Work package 4 and work package 5, these are both our research and clinical federated applications. So there's a whole set of quite interesting applications we're looking at, things like federated GWAS, federated joint genotyping, diagnostic services, and various fair data analysis applications. And then obviously we're there's quite a significant ELSI. We got work package 7 and 9 are both supporting, looking at the whole ELSI questions across this tri-continental scenario of cohort access. And obviously work package 6 is our outreach and training, and work package 8 is our management and coordination. I think the point that we realized very early on in the project is that even if you took out any one of the work packages here, there's this real interdependency between all of our work packages. If you removed any of one of these work packages, the project couldn't succeed, essentially. You have to have your federated data discovery to be able to find the cohorts of interest. You couldn't do your clinical research applications if you haven't been able to find the cohorts that are relevant and have the right metadata for your application. You couldn't do, if you don't have the AI interoperability to be able to do this kind of loop of requesting access and getting access and identifying yourself, you really wouldn't be able to do these federated applications if you didn't have that from work package 2. And the same goes for almost all the other work packages. So there is a really kind of tight dependency, and I think we realized this early on in the project, where we set up these kind of cross work package working groups where we would sort of focus on a particular research or clinical application and say, well, I need this metadata to be harmonized for work package 3. I need that metadata then to be discoverable through the work package 1 data discovery mechanisms. And I need to be able to have the AI interoperability between these particular cohorts. So, I think we realized, we addressed this kind of interdependency by just said we had to essentially set up these cross work package kind of focus groups. Where we are on the project, so I think early on it was the sort of more sort of technical work packages 1, 2, and 3, the data discovery, the metadata harmonization, and the AI interoperability. They were really very active in the first half of the project, and I think the focus really now is on these developing our federated research and clinical applications. I mean, building on basically what work package 1, 2, and 3 have achieved. And then we have the other work packages, the outreach and ELSI and project management running throughout the whole project. So I'll just try and give some brief examples of, and I'm not a fair expert, but I'll try to give you some brief examples of, how we have, if it's not sort of obvious already, but how we have tried to address the sort of increase the fairness of the cohorts participating in the CINECA project. So findability, I think it should be quite obvious. I've talked about it certainly in the last few slides. I mean, this is our work package 1. It's the very first problem. How do you find cohorts of interest? And I think, what we've tried to do is we've tried to break it down to a kind of logical set of different mechanisms or ways of how you would want to find cohorts. So, you can break this down into different query types. I mean, you've got uniform queries that should be span across all of the cohorts, like what are the number of participants you've got in your cohort, maybe what types of data have you collected, what assays have been carried out across the cohort, these kind of uniform queries. Then you get into the extended queries, which are maybe going much more in detail into the metadata, the cohort sort of phenotype data, and really rely on work package 3 to have done the kind of harmonization, the cross mapping between the phenotype and metadata fields. And when I say that, I mean, what I mean essentially is, here's an example here from this is the Gecko ontology is essentially the model that we're using to cross map between our cohorts, and how you link basically from a particular cohort. So, you could be collecting the same metadata field about, relevant for the same disease, but it's just got a different naming or a different ontology field, and be able to just do this cross mapping. So we're cross mapping all of our cohorts into this Gecko ontology, and then using that essentially as the way of doing the mechanism to sort of drive these more complex queries. And actually, we've been in recent work, we've been using this same Gecko ontology with the IHCC to do cross mapping between the IHCC cohorts to enable to drive the IHCC cohort discovery atlas. And then there's various other aspects here. There's a service catalog about finding out, what are the various API endpoints that is possible to query for a particular cohort and what's sort of available. And this feeds into the GA4GH standards around service catalog. And then ultimately we want to enable these federated queries. So if we want to be able to link into networks like, like the Elixir beacon network, like the GA4GH beacon network, to be able to send out sort of queries, and the Candig network as well. I mean, to be able to send out sort of federated queries to be able to get back those discovery queries and get back the responses from the various cohorts. Okay, I think I've covered this. Yeah, I've talked about Gecko. Yeah. Okay. So next thing, thinking about accessibility. I mean, I'm only picking specific examples here, but there's probably many more examples you could pick from the project. But in terms of accessibility, I mean, I guess, we have this work package 2, which is all about sort of getting access to cohort data. And what the challenge we're trying to address here is trying to make the authentication and authorization systems to be interoperable. We did provide one of the very first implementations of the GA4GH researcher passport standard in early 2020. And what we were trying to do here essentially is get linkage between the Candig AAI. So a researcher who logs into Candig can present whatever their credentials are. I've got access to these data sets and these cohorts, and then be able to sort of exchange that through the Elixir network to be able to identify themselves, perhaps if they go to apply for access at another cohort, so that they can have sort of this digital kind of passport essentially that identifies that researcher. So we have got linkage between the Elixir AAI talking through GA4GH passports, and also with the EGA. And I'm hoping, maybe, well, Nikki can comment, but I'm hoping we will be able to get linkage between the H3 BioNet at some point, maybe this year or next year, with the EGA in terms of this data access and authorization loop. Yeah, and then I would say, our work package 4, this is around our research applications. They're looking at implementing some of the more novel data access protocols. I mean, the example here is the GA4GH HTS GET secure streaming service is one of that's some of our cohorts have implemented. I think the point is that we have to be realistic in that I'd love if every cohort participating in CINECA will be able to implement all of the different fair standards that we're implementing to increase the fairness. But we have to be realistic about this sort of, in terms of the funding that's available and what's possible. So we're trying to be as comprehensive as we can within the boundaries of what we have in our funding. In terms of interoperability, I mean, fundamentally, that is what the goal of CINECA is to make the participating cohorts more interoperable. And I hope through the various work packages that is happening, be it on the metadata level, be it on the accessibility and AAI, be it on the discovery queries. I mean, this is really is the fundamental goal. So we really, when we were thinking about and designing the CINECA project from the very start, we made a really clear alignment between CINECA and GA4GH, so that it became almost like a way of sort of driving the GA4GH standards in terms of, and also deploying them at scale and seeing if they're fit for purpose, and then feeding that back into the GA4GH standards process. And I think we've certainly have been quite actively doing that sort of loop throughout the CINECA project. Interoperability, I guess, on the metadata level. So we have this Gecko ontology, which is now mapping, probably mapping between, it's mapping between the participating CINECA cohorts. We've also now extended this to we've started working with the IHCC and their cohort discovery atlas, and using Gecko again as a way of linking between those various cohorts, the data dictionaries from those cohorts. So I think this has really been certainly a success story in terms of the alignment between CINECA and the IHCC, and using the same sort of scheme for doing this metadata mapping, while supporting the diversity that there is between the different cohort metadata collections. And then finally, we've been trying to really promote the use of the data use ontology, so that cohorts can really upfront, sort of on their website or on wherever it is the public facing location, essentially, for the cohorts, be it an API or on the website, to be able to annotate their cohorts with this data use ontology, so that researchers can see up front, what are the sort of appropriate types of research that can be carried out on those cohorts. And if we're using, of course, if we're using the same ontology for this, it should be much clearer and easier to be able to answer that question very early on. Yeah, I talked about the IHCC. This has been a really, extremely positive collaboration that we've had between the CINECA project. I mean, they're aiming to create this global network for translational research that, utilizing large cohorts to sort of understand, to enhance the understanding of the biological and genetic basis of disease. So the goals of IHCC are very well aligned with CINECA. So we've had a really positive collaboration through CINECA Work Package 3, in particular. Melanie Courteau has been leading that effort. Fundamentally, the link that we have is that we're using the same ontology or same mapping model for mapping between cohort, the cohort and metadata and phenotypes. And you can see now on the IHCC cohort atlas is using this ontology now as a basis for their cohort discovery portal. It's still small scale. It's only got, I think, maybe less than 10 cohorts in there at the moment. But we hope that this is going to build and extend into future years. And I think that the CINECA project has really got this process going, I think. Yeah, I mentioned international standards again. We really want CINECA is meant to be a driver for those standards, identifying where there's gaps in the standards, showing how they can be implemented at scale, feeding back, having a sort of feedback loop to those standards to be able to highlight issues or really show where they work or where they're not working. And I think we can identify throughout the various work packages, there's multiple different standards that are being implemented at the cohorts, which is really really what we want to do, essentially, to increase the fairness of the CINECA cohorts. Yeah, and I guess reusability. In terms of reusability, I mean, ultimately, one of the core sort of measures of success for many of these cohort infrastructures is to really show that their cohorts are being reused by researchers throughout the world. I mean, and that's obviously that's a core metric for many of the funders of these cohorts to, to be able to demonstrate that these cohorts are adding value and are being reused for research. So, I think it will probably won't become, the impact of CINECA probably won't really become sort of visible in terms of cohort reuse. Maybe it'll be on the end of the project, or maybe towards the end of the project. But we really do expect that, by implementing these various ways of implementing the FAIR standards, I think we really expect that that these cohort data will be more widely discoverable, queryable, accessible, and hopefully ultimately reusable. And to really accelerate the research and clinical applications that would not have been possible otherwise from our participating cohorts. Okay, and then so I guess one way that we're going to try and demonstrate the use of and reuse of the cohort data, is really by having our how would you say, demonstrator federated research and clinical applications through our work package 4 and 5. We are really trying to show that this federated analysis paradigm is the correct paradigm, but also we really can answer some really convincing and relevant research and clinical questions within this federated analysis paradigm. So, there's on the right-hand side of my slide here, there's a schematic of how we're proposing to do the federated joint genotyping. That's one of our particular applications, and how we're actually going from what that might look like in a federated paradigm to really kind of turning it into how does it actually work at the kind of logistically, what technologies we would use, which standards we're going to use, what protocols, what are the particular cloud environments that application will be run over. And I think we've got so that's just one of them. We've got a whole set of other applications. So there's the federated polygenic risk score analysis, genetic analysis of sequence-based phenotypes, and then we've got our clinical applications. So like the FAIR data analysis for cancer biomarker discovery, and analytical sandbox for diagnostic services. And it's really in the second half of the project that these particular applications are really kind of the main focus, because it's they're building on what was the work that was carried out in the first half of the project. So I think we're starting to see some of the demonstrators coming from these applications, and it's really quite cool to see some of these federated applications really happening, really sort of being developed and being demonstrated in this, and building on what was carried out earlier in the project. I did mention the ELSI landscape, and it's something that we can't forget, because it's it is certainly a significant challenge, especially with the sort of regulations and that have changed in the last few years, and I'm sure will continue to change how we understand and interpret some of the national laws that are potentially governing how data can be cohort data can be accessed and made available, and this is a constantly changing environment. And I think one of the, I've just highlighted here on the right, one of the particularly interesting deliverables that was written from our ELSI work package, and this was really kind of looking at the sort of the different regulations that operate between sort of Canada, Europe, and Africa, and trying to look at the legal gaps, trying to look at the differences between the regulations. And it's a super interesting report to look at. I think it is available on Zenodo somewhere. It's super interesting because it really looks at, like, what are the differences between the GDPR regulations and the corresponding Canadian regulations. I think there's a lot of emerging sort of policies and regulations coming from through the history of Africa, and starting to look at how do they compare then to some of the regulations that are already in place in Europe and Canada. So it's a super interesting report to read. And then finally, I mean, ultimately we of course want to be working with the actual cohort data in itself. I think we realized that I guess maybe 6 months into the project, that if all the cohorts were totally fair and you could very rapidly get access to cohort data, and everything was working really well, and you weren't sort of having to wait sort of the sort of maybe even perhaps months to get access to cohort data, then perhaps we could work with we could be able to use the real cohort data for all of our sort of research and clinical applications. But I think we realized quite early on that we really do need to have some some sort of test or synthetic cohort data that we can start to build some of our demonstrators on, because it's unencumbered by the sort of the pre-authorization required for getting access to the cohort data. It can be more openly shared. It doesn't have like any sensitive information, any personal information in there. And I think we have had a working group that's just about completed, I think now, that's developed a whole set of synthetic cohorts that are modeled on the real cohorts. So, there's a synthetic cohort that's been developed that's modeled on sort of a combination of the 1,000 genomes and the UK Biobank data dictionary. Doesn't use any of the real sort of individual level values. And there's a variety of other synthetic cohorts here that are now available. They've been loaded into some of the various systems that we have, the discovery and the data access. So you can actually really use these cohorts to really start to build some of the federated applications using the exact same sort of interfaces and standards that we're implementing with the actual cohorts themselves. And I think this has really been, I think we realized quite early on we really need to have this in place so that our research and clinical applications can really start to build their demos without having to worry about sort of sensitive data and then doing, if they have to wait quite a while to get access to the real cohort data. So these are available now, and yeah, I think there's been quite a lot of interest in these synthetic cohorts, and I think it's an area that other projects like the B1MG and the 1+MG in Europe project, the Million Genomes project, are quite interested in this, in having these kind of synthetic cohorts. That's another sort of, again, another cross-cohort, international sort of looking at international accessibility of human genetic and phenotypic data. That's everything I had. So here's a picture of, we did actually manage to have one physical meeting, the kickoff meeting for the project, and then, well, everything else happened, COVID, and we have never managed to have another physical meeting, but maybe we will before the end of the project again. And yeah, I've tried to give some sort of summary of the project. There are, all of our work package leads and individual groups and working on their individual tasks are the real people really doing the actual work here. So yeah, please, if there are any detailed questions, I'll probably redirect them to some of the more experts in all these kind of different areas in the project. And happy to answer any questions.
Great, thank you, Thomas, for a great talk. We know that what you've demonstrated is that actually, without being fair, CINECA won't work. It entirely relies on being findable and interoperable, and of course reusable for a federated discovery to actually work. So the floor is open for questions. You can either raise your hand or you can type in the chat. So please feel free to do either one of those. We'll kick off actually there were two questions that came in by email during the registration. The first one is actually from Kirs. I don't know if you want to just ask a question. He asked how are persistent identifiers for datasets, genes, persons, files, people, organization, etc. minted or used or selected in the context of CINECA and the GF4GH standards?
Yeah, fair. It's a really good question. Fair question. I think one of the approach that we've taken with the CINECA project is that we're not trying to build like a CINECA system or a CINECA platform. The whole approach we've taken with the project is to try and enhance the existing sort of infrastructures, existing systems. I mean, the example being, say, the Elixir AAI. Rather than trying to build our own CINECA AAI system, and then having the issues about sustainability beyond the project, we're trying to sort of contribute to the existing infrastructure. So coming back to the question, I suppose the approach we've taken is that there are participating cohorts, there are participating cohort resources, like the EGA, that have existing identifier schemes that are set up and recognized by the community. And I guess we want to sort of use those existing schemes rather than creating, I guess, creating a CINECA identifier scheme and then having the sustainability issues in the future. So we're really relying on the existing mechanisms that are from the various participating resources. So be it CANDIG, be it EGA, be it, I don't know, through Africa, maybe you assign identifiers as well. But it's really building on the existing schemes that are out there rather than trying to invent our own identifiers and yeah.
Great, thanks. Kes, I hope that answered your question. So another question came in from Etienne. He asks, are there any plans to also include the CARE principles for Indigenous data governance? Provides a link to CARE. I don't know, do you know about the CARE principles?
I hadn't seen this actually. I hadn't heard of it before.
I'll paste in the link.
So it's yeah, I hadn't heard of it before actually. Those principles, which is interesting. I mean, I know.
So, sorry if I may quickly jump in. I asked that question. So the CARE principle stands for the collective benefit, the authority to control, responsibility, and the ethics. And these are principles like that have been set up by the Global Indigenous Data Alliance. And certainly in Canada, this is becoming increasingly important to consider those aspects. And I'm sure that it would be really interesting to see also how that could be applicable in Africa. Maybe a little bit less so in Europe because there's fewer such populations, but yeah, no, it's overall very interesting, and it's not exclusive of the FAIR principle. They're meant to go actually hand in hand, but just slightly different. So
does it then consider sort of sharing of data more than anything else in terms of ethics and protection? I'm just looking at the website now, but we'll certainly look at it from the African perspective. Etienne, that was a question to you.
Sorry, it is really about data governance, and actually it's a collective ownership of data, and the fact that it's not necessarily to block that, but that the authority to access the data must be governed by Indigenous people after evaluating, like from an ethic perspective, if there's benefit for their communities as opposed to like just research or other communities or individuals. So that's that's kind of the space in which that has been created. And there's certainly been a lot of burned bridges, I would say, while in North America, with various Indigenous populations, that their genomic data has been misused or used in ways that were not hadn't been approved. So for us, actually, to include Indigenous genomic data in our analysis, it's something quite crucial to be aware of that and to build trust with those communities. Really, the whole idea being to reduce the genomic divide that can exist as we know more and more of certain population, like Caucasians, but less so, let's say, from other populations that might be more reluctant to embark on cohorts or gene discovery activities.
Okay, so definitely something that should be considered in CINECA. Thomas, did you have any comments on that? Or
no, I mean, I guess, yeah, I guess given the sort of funding source that we have with CINECA, it is largely dominated by the sort of population of cohorts for this, the kind of ethnic origin that you usually see, the sort of bias you normally see. I think, I mean, one thing I would say is that, you know, the IHCC, which is the other, you know, a kind of similar project that's really kind of in an earlier phase, perhaps, there is a real focus on trying to have, you know, better sort of representation of and diversity of across the cohort, so that truly is sort of more global than any of the existing. Well, you know, most of the existing cohorts have this Caucasian bias essentially, and I think you know, I think that is a real focus at the IHCC, and we talk about it all the, you know, at this sort of steering committee groups, we talk about this all the time, that we really need it to be representative of have representation of the underrepresented populations potentially that have not had, you know, as good representation in cohort studies in the past.
Yeah, I think a lot of these initiatives are bringing up this increased diversity and inclusion, but it does come at not the cost, I don't want to say it's the cost, but having to consider these additional issues around Indigenous populations. So are there any other comments on that or some questions? So Melanie's put a comment in. She thought it was brought up at the G4G's regulatory and ethics, particularly with respect to the data use ontology of Australian colleagues. Yeah, because they work with Indigenous populations as well. Okay, so yeah.
So I was just gonna say the discussion around benefit sharing and things like that, those are really interesting. Those are discussion we're having as well with respect to the data use ontology. Thomas was talking about, we have a request to anchor geographical restriction, but then some participants say, well, does that mean that the population that contribute genomic data benefit from results derived from that genomic? So there's really an interesting topic around here, and probably something that H3 Bionet and CINECA and G4GH could be involved in.
Yeah, certainly in East Africa, there are a lot of discussions about benefit sharing, and in the context of commercialization and partnering with commercial entities. You can't really get a product without doing that can benefit the population. And so there are lots of these. So these are all ethical discussions, but I think it does impact fairness. So when I talk about fairness in East Africa, people assume that it means we're going to make everything open, but it's not. It's just making information about how open it is and how to access it when you go through the right channels. So there's also a bit of education there with these different more sensitive population groups to make sure that it's the understanding of fair for their data does not mean completely open. It means just providing more information about that data set itself. Which actually brings me to a question that I was thinking about when Thomas was talking about Duo. Does that come under R, reusability, or does that come under interoperability? Access, sorry, because it's describing what you can do with the data, but it's also how you can reuse it, or is it really just both? I mean, maybe I'm nitpicking here, but what we're finding from the discussions on the hackathon is that there's quite a lot of overlap between the FI, AI, and R sometimes, and you don't know which one of those some things go into.
Oh, if you get to tick two of them with something, that's okay too.
Yeah.
Yeah, I had this exact same debate when I was thinking about, like, how do I match different parts of what we're doing in CINECA to which one of the letters, and yeah.
I was gonna say we should tick the four. So an interesting thing that with Duo is with respect to the findability. So we've had a report, for example, from the Sangha, who was pioneering the annotation of data sets with geoterm. They said they've noticed that people actually search using the geoterms, and their data sets are found more often because they have those annotation. So I think so that's findability. Accessibility, well, it does describe interoperability, because you can take data sets from North America, Europe, Africa, and it's a shared standard. And reusability, we need to find a good argument for reusability to tick the fourth box.
Well, it's saying what you can reuse it for.
Yeah, it says what you can do for secondary data. So yeah.
Yeah, so it does actually tick all the boxes. Yeah. Good. Are there any other questions for Thomas? So you can paste them in the chat, or you can raise your hand. I haven't seen raised hands. But feel free to raise your hands in the last few minutes or to type a question. I just have one more comment about, Thomas talked about making CINECA fair. In the hackathon, we're talking about very specific things like cohort data, training materials, and software. Where CINECA is a project, and actually I think it's the pieces of CINECA that are being made fair, like the individual cohorts, and maybe the software within CINECA. I mean, there's no metric for saying the CINECA project is fair. I guess you can only say pieces of the CINECA project which make it feasible and successful are fair. I mean, would that be a reasonable comment, or do you think actually you can say CINECA as a whole, as a project, is fair?
I think, I think it's when I talk about fair now, it's more about how we're implementing fair in CINECA, and how it maps into all the different work packages, and it's how it's making, hopefully, the participating cohorts more fair. But there's so many different aspects to this, and it's been tackled in so many different ways across the project. I mean, I don't know if you can say CINECA is fair, but it's what we want to do is make the cohorts more fair, or increase the fairness of the cohorts, and that's fundamentally, if you were to write down what is the fundamental reason or sort of question behind CINECA is how can we make these cohorts fair in a federated sort of context, and that's and just there's so many different aspects to that, and so many different technical parts to that, and so many different as we've heard throughout the getting this kind of overview of the project. There's so many different ways in which we do that. Fair is complicated.
Yeah, but I also found I had a discussion with a biochemist the other day, and I said, we were talking about fair. They said, well, what is fair? They'd never heard of fair, and I was staggered. I thought the whole world had heard of fair by now. But I think also, educating people as to the data users rather than the data people as to what is fair. And I think CINECA is a good way to demonstrate that if things are not fair, you can't actually do cross-consortium analyses to help health, to benefit health. So I think there's a lot of education that needs to happen with examples like this of the non-data people, the non-data community who are completely afraid with fair, I think, by now, but it's the non-data people who don't understand the benefits.
Well, showing basically, I mean, this is why we have these research and clinical applications, is to show what's possible, by making some of our cohorts more fair. What can you actually really do when it really matters, when it comes to trying to do whatever your research application is. I mean, and we've got these exemplars essentially where we're trying to show, look, this is what you can do now that you couldn't do before. And the scale of this challenge is huge. I mean, to make the world's cohorts fair and using standards. I mean, this challenge will go on for many years, and it's certainly we're trying to move the dial forward with some of the emerging standards and show how they work or they don't work, and ultimately try to make the cohorts that we have participating more fair.
Yep. Okay, so if there are no further questions, then thanks very much, Thomas, again for the webinar, the final one in the series. So that'll again, the recording will go to YouTube with the other series of webinars. I did paste in